Hello everyone, my name is AJ Keefe, and in this presentation I'll be reviewing DNA-PK, a DNA repair protein, and HSP90, a critical folding chaperone. Using these two concepts, I'll then cover this paper titled DNA-PK promotes mitochondrial, metabolic, and physical decline that occurs during aging, published in the Journal of Cell Metabolism. I recommend that my viewers pause the video at each new slide and read through the material and analyze the graphics before playing my commentary. And if you have any questions, please comment below and I'll promptly respond. Thanks for watching. DNA-PK is a DNA repair complex composed of a scaffolding protein, heterodimer, of Cu70, you see right here, Cu70 and Cu80. And also a catalytic kinase, DNA-PK-CS. So what this complex does is it essentially scans for double-stranded DNA breaks using this Cu70, Cu80 domain. And upon finding one, DNA-PK autophosphorylates itself and it becomes an active kinase. In the canonical pathway of DNA repair, this enzyme functions to recruit various DNA repair enzymes and mediates the process of non-homologous end-joining, or NHEJ. NHEJ is simply an error-prone mechanism of DNA repair that is actually the primary method of DNA repair in post-mitotic cells like neurons. And the reason post-mitotic cells preferentially repair DNA damage through non-homologous end-joining is because without chromosome replication, the homologous template needed for homology-directed repair is absent. And instead, the cell has no choice but to use non-homologous end-joining and to simply digest the broken DNA and then ligate it together. And then kind of basically hope that it didn't occur in a critical gene. The loss of DNA-PK actually causes a neurological disorder because post-mitotic cells like neurons are unable to efficiently repair their DNA. And they also have a severe immunodeficiency phenotype because VDJ antibody recombination, which produces various antibodies, actually requires this faulty DNA repair process of non-homologous end joining in order to make those antibodies. So the kinase activity of DNA-PK is actually not confined to DNA repair. We can see, although it's interacting with ATR and also phosphorylates ATM, it phosphorylates many DNA repair proteins, but it's not confined to DNA repair. And it's known to phosphorylate various cytoplasmic and nuclear proteins as well. So although it is DNA dependent, it can phosphorylate many other proteins that are not necessarily related to DNA repair. And one of those proteins that is phosphorylated by DNA damage activated DNA-PK is HSP90. I have that added in this figure. HSP90A, or alpha, is phosphorylated by DNA-PK. And HSP90 alpha is a folding chaperone that we'll discuss in the next slide. But currently, the functional consequences of DNA-PK phosphorylation of HSP90 at positions 5 and 7 is unknown. And so that's one of the topics of this paper, is to define or to figure out what's going on with this HSP phosphorylation that occurs during DNA damage. So DNA-PK phosphorylates HSP90. What is HSP90 and why should we care about this phosphorylation? Well, HSP90 is actually an incredibly important and evolutionarily conserved protein that functions to simply fold proteins that are recognized by aberrantly exposed hydrophobic residues. So this other protein, HSP90, is actually the initial protein that recognizes these aberrantly exposed hydrophobic residues on proteins that should be folded within the hydrophobic core of the protein. And if it's exposed to the cytoplasm, this HSP70 binds it and actually passes these proteins on to HSP90. You can see that in this graphic as well. So HSP90 is so important that we actually have 5 isoforms of HSP90. And although they're each a little bit different in their function and their tissue distribution and subcellular localization, they differ most importantly by the proteins that they interact with. The proteins that interact with a folding chaperone are called client proteins or clients. And HSP90 alpha is the isoform that is phosphorylated by DNA-PK, and it's known to modulate the folding of over 700 so-called client proteins. Additionally, the proteins HSP90 alpha appears to preferentially interact with are commonly kinases. So HSP90 will bind to either newly translated proteins. So as it's being translated out of the ribosome, HSP90 is there to catch it and to fold it as it's coming out of the ribosome, or it will simply bind proteins that have spontaneously misfolded, long after translation. And HSP90 actually typically receives these misfolded proteins, again, from HSP70, which is what initially binds the hydrophobic residues. And so HSP70 hands off these misfolded proteins to HSP90, and preventing this handoff between HSP70 and HSP90 will shuttle the misfolded proteins into the UPS, or the ubiquitin proteasome system down here. So this is typically carried out by UBR1, COL5, and I think CHIP. CHIP is probably the most important E3 ubiquitin ligase that associates with HSP90. And inhibiting the proteasome ubiquitin system typically leads to aggregation of the proteins. So in other words, the inability of HSP90 to bind its clients results in their degradation via the proteasome. If we prevent this handoff right here between these two proteins, if you inhibit that, it turns into either degradation, or if the proteasome is overwhelmed or saturated or inhibited, it leads to aggregation. And that's really huge, because that it basically means that if something were to happen to HSP90 right here, if something were to happen to its ability to function, some 700 proteins will be entering the UPS instead of getting folded, or potentially misfolding and forming aggregates, such as amyloid plaque or something. So HSP90 knockouts are a good way to illustrate the phenotype of and what actually happens when HSP90 is knocked out. So some of the HSP90 knockouts in Drosophila would have deformed wings, and another will have a deformed brain, and even other HSP9 knockouts might have deformed antennas. And the reason a single protein is able to have so many different phenotypes is because if any one of HSP90's folding clients happens to have some point mutation, it might be uniquely prone to misfolding. For example, if a brain development protein has some common mutation, say it's 20% of the population has this mutation, it might normally get away with this mutation when HSP90 is present, because it will still get folded properly. So the mutation might promote misfolding, but as long as HSP90 is around, it still folds correctly. But the second HSP90 is lost, through deletion or through spontaneous mutation in the genome, this point mutated brain development protein will then begin misfolding. And this idea is known as evolutionary capacitance, where mutations do not produce a phenotype until their associated folding chaperone is lost. Eventually, the evolutionary potential kind of accumulates, but it's kept in check by the folding chaperones. But it's all lost when HSP90 is deleted or is mutated, and it just basically causes all protein hell to break out in the form of protein misfolding. So keep this in mind as we discuss what happens to HSP90 during aging. Because during aging, I'll give away the ending, HSP90 activity decreases. So as HSP90 activity decreases, this might cause misfolding in various, some of the 700 proteins that are serviced by HSP90. And so this could cause a very wide range of phenotypes, kind of like the wide range of phenotypes that we see in aging. So it's possible that HSP90 activity downregulation that occurs with aging could be causing the the pleiotropic effects of aging as well. Anyways, you can see in this graphic that the addition of an HSP, you see down here you can see HSP90 inhibitor. The addition of this HSP90 inhibitor, such as 17-AAG, which we'll see in this paper, causes the degradation of client proteins. So the loss of HSP90 triggers degradation of client proteins. AMPK is a very important HSP90 client. In fact, it's not just AMPK, it's also its upstream activator, its upstream kinase. Liver kinase B, or LKB, is another client of HSP90. And these two proteins, AMPK and liver kinase B, they function to promote mitochondrial biogenesis in response to starvation. And AMPK is a hugely important kinase, maybe even my own personal favorite kinase, because what it does is it allosterically binds to ADP, and when it's then phosphorylated by its upstream kinase, LKB, liver kinase B, it initiates a potent starvation program. And some of the downstream effects of AMPK activation include mitochondrial biogenesis, autophagy induction, translation inhibition, and many other things. So for the purposes of this paper, just think of AMPK activation as synonymous with mitochondrial biogenesis, and it is generally a very good thing to activate. AMPK is generally good for the cell. And since HSP90 services AMPK and assists in its folding, as well as its upstream kinase, LKB, we can say that HSP90 is upstream of AMPK, because without HSP90, AMPK wouldn't get folded, and it would just get degraded by the proteasome. So this would presumably lead to its degradation if HSP90 is inhibited. So keep that in mind as we discuss what this paper discovers. So we have three basic ideas at this point. First, DNA damage that accumulates with age activates DNA-PK. Second, DNA-PK phosphorylates HSP90. Third, HSP90 assists in the folding of hundreds of proteins, including critical proteins like AMPK. So given these three assumptions, it might then seem plausible that DNA damage-induced activation of DNA-PK could be modulating the folding of AMPK through its phosphorylation of HSP90. And so this is what they're going to be looking at in the current paper. So first they confirm that DNA damage increases with age, kind of simple, and this leads to a corresponding increase in DNA-PK signaling as the organism ages. And this also leads to increase HSP90 phosphorylation at threonine 5 and 7. So as we age, DNA-PK signaling is picking up, and in one of its substrates, HSP90 at positions 5 and 7 of threonine increases. So the DNA damage-induced phosphorylation of HSP90 is discovered to prevent it from binding to its various client proteins, including AMPK. The inability for phosphorylated HSP90 to bind client proteins leads to the downregulation of its client proteins. And knocking down or deleting DNA-PK decreases the amount of inhibitory HSP90 phosphorylation and subsequently prevents DNA damage from downregulating their expression. Preventing HSP90 phosphorylation by mutating the threonine 5 and 7 also prevented DNA-PK from downregulating client protein expression. And finally, the authors observed that downregulation of AMPK due to HSP90 phosphorylation and inhibition leads to widespread metabolic dysfunction. So the idea here is that DNA damage accumulates with age. We can see this right here. DNA damage is accumulating. This is activating DNA-PK signaling, which leads to HSP inhibition through this very specific phosphorylation. And this causes critical signaling proteins like AMPK and liver kinase B to be inhibited, to be downregulated. And this downregulation of AMPK disrupts things like metabolism, fitness, and leads to obesity as well. The researchers first wanted to examine if there was an age-dependent increase in DNA-PK or HSP90 phosphorylation. And they found that in muscle cells, as DNA damage increases, as measured by this H2AX immunostaining, so we see in these old muscle cells, we see increased H2AX indicating DNA damage. See, as this increased, so did these activating DNA-PK phosphorylations. In these Western blots, we see increased staining of P DNA-PK phosphorylated. We also see increased HSP90 inactivating phosphorylations. So this is in graphical format. We can see it very clearly. The amount of phosphorylated HSP90 increases by about 4 or 5 times. And this is an antibody that's specific for the threonine 5 or 7, which is inactivating. We also see phosphorylated activated DNA-PK also slowly increasing with age as well, which makes sense because DNA-PK is binding these gamma H2AX double-stranded breaks. If HSP90 is being phosphorylated by DNA-PK, then it needs to be in the nucleus because DNA-PK is a kinase that is activated by binding to double-stranded DNA. And so on the right, we see they did some cell fractionation. So they separated cytosolic and nuclear fractions. Then they did this Western blot to see exactly where HSP90 was found before and after irradiation. So they're doing post-irradiation. Is it in the cytosol or is it in the nucleus? And we see that there's increased nuclear translocation by 4 hours. We see there's a lot more phosphorylated and total HSP90. And we also see it in the cytosolic fraction. So in general, there's more HSP90 and there's more phosphorylated HSP90 in both the cytosol and the nucleus. On the left, we see the quantification of HSP90 interaction with LKB. So this is a co-immunoprecipitation. So they're pulling down liver kinase B1 and they're staining for HSP90. The interaction between HSP90 and its folding client LKB1 decreases following DNA damage. So we see in this wild type condition when DNA damage is present right here in these lanes down here, the interaction between HSP90 and liver kinase B is decreased because there's decreased staining. However, if HSP90 is mutated at residues 5 and 7 right here, the interaction between liver kinase B1 and HSP90 persists through DNA damage. So this mutation prevents it from being phosphorylated, and preventing phosphorylation allows the interaction with its folding clients to persist even after DNA damage when it's normally downregulated. And in this other lane right here, we're seeing what happens when HSP90 residues 5 and 7 are mutated to what's called a phosphomimetic, which is basically a residue that looks like a phosphorylated amino acid. We see that when HSP90 looks phosphorylated at positions 5 and 7, there is constitutive downregulation of its interaction with the LKB1 folding client. So even without irradiation, it's still not binding to its liver kinase B1 folding client. So when it's phosphorylated, it's not interacting with liver kinase B1, its folding client. So far, we've been shown evidence suggesting that DNA damage induces the phosphorylation of HSP90, and this phosphorylation decreases its association with client proteins. On the left, we see another instance of HSP90 interacting with a client protein, and blocking phosphorylation at 5 and 7 again increases the co-immunoprecipitation between HSP90 and AMPK. So this is what we saw on the last slide with liver kinase B. We're seeing the same thing but with AMPK. So its association increases when it's no longer able to be phosphorylated at these two residues. So the question now is, what is phosphorylating and inhibiting HSP90 during DNA damage? The specific kinase is still not pinpointed. We know that DNA-PK is capable of that phosphorylation. We don't know that's the sole reason it's happening. Well, when DNA-PK is knocked down with siRNA, we see in this middle lane, when the siRNA is applied, we see that the interaction between HSP90 and its client protein GR, or glucocorticoid receptor, is able to remain. So I'm not exactly sure why they used the GR receptor instead of just HSP or AMPK. I think that would have made more sense. They said something to the effect of the interaction isn't as observable enough. But GR is a well-validated client protein of HSP90. And we see that in control conditions following irradiation, the interaction is very, very much disrupted. There's almost no pull down or co-immunoprecipitation between HSP90 and the GR. But when siRNA towards DNA-PK is administered, the interaction persists. So inhibiting this single kinase, DNA-PK, allows HSP90 to interact with its client proteins following DNA damage. Additionally, on the right, we see that DNA-PK siRNA significantly downregulates HSP90 phosphorylation. So we see that post-radiation, 2 and 4 hours post-radiation, when DNA-PK siRNA is present, we see almost no DNA-PK, obviously. We see very little phosphorylated HSP90, but we still see adequate levels of HSP90. It's just not phosphorylated when DNA-PK is knocked down. So DNA-PK is the primary kinase responsible for phosphorylating HSP90 at positions 5 and 7. To confirm their findings in a live animal, they generated a muscle-specific knockout of DNA-PK right here, these MDPKO cells. And these are going to be the white bars in these graphs. And they found that these mice showed an age-dependent decrease in the amount of phosphorylated HSP90. So we see the amount of phosphorylated HSP90 at 17 months is, only 30% or 20% of what it was in the normal control cells. So when DNA-PK is missing, HSP90 is not getting inhibited at old age, which is presumably a very good thing because inhibiting a folding chaperone in old cells is probably not a good life decision. And so this relief of HSP90 inhibition is also accompanied by increased phosphorylated and activated AMPK. So again, down here we see AMPK, which is promoting mitochondrial biogenesis, is increased when in these MDPKO cells that are lacking DNA-PK. On the right, we see more work in muscle cells. So we're going back to muscle cells. We're knocking down HSP90 in these two graphs. We're knocking down HSP90, and we're seeing a huge decrease in AMPK. So when HSP90 is missing or inhibited by DNA-PK, it would cause decreased AMPK expression. We also see these graphs are coming from a Western blot, such as what we see here, and we see again that siRNA towards AMPK knocks down, or siRNA towards HSP90 knocks down AMPK. And finally, we see up here in the corner that this 17-AAG, which is a very specific inhibitor of HSP90, this inhibiting HSP90 is able to decrease AMPK expression. So when HSP90 is inhibited, we see decreased AMPK expression. And when DNA-PK is deleted, we see decreased inhibiting HSP90 phosphorylations. Okay, so what consequence does this DNA damage, DNA-PK, HSP90 inhibition, AMPK downregulation pathway actually have? Well, on the right, we see that there's actually a pretty impressive inverse correlation between the amount of mitochondria and the amount of phosphorylated DNA-PK. So in these aged rhesus macaque muscle cells, as the amount of activated DNA-PK increases, so down here, as we see more activated DNA-PK, we see decreased mitochondrial biogenesis, or mitochondrial, density. So to suggest a mechanism for this other than AMPK modulation, they looked at PGC1 alpha down here, which is a master mitochondrial biogenesis transcription factor. And in these MDPKO DNA-PK knockouts down here, we see that the expression of PGC1A is vastly upregulated in aged mice. So a master mitochondrial biogenesis transcription factor is upregulated when DNA-PK is knocked out. And additionally, at the whole organism level, we see that mice harboring a DNA-PK mutation in these so-called SCID mice. It's a well-known mouse that's used to mimic or to model immunodeficiency because it's lacking its DNA-PK protein, which is needed for VDJ antibody recombination. Anyways, in these mice that are lacking DNA-PK, over here on the left, these SCID mice, we see significantly increased fitness judged by their running distance, and it's actually pretty significant in these MA or mature mice. These older mice, we see very significantly increased running distance. We also see increased running distance when DNA-PK is inhibited. So in these white bars, there's an inhibitor of DNA-PK that is present. And when DNA-PK is inhibited with a small molecule, we see these animals are, again, much more fit, and they're running longer distances. And lastly, this DNA-PK's inhibitor's ability to increase animal fitness is dependent on the presence of AMPK. So this is actually a really important figure right over here. And it shows that when when AMPK2 is knocked out, so in these lanes, this is AMPK. When this protein is knocked out, which is a muscle-specific AMPK, the benefit conferred by these DNA-PK inhibitor is lost. So this suggests that DNA-PK inhibition increases fitness through the AMPK pathway, because when the AMPK pathway is missing or disrupted, we don't see the benefit of inhibiting DNA-PK. So the summary of this paper, this is the general pathway. So we saw DNA damage activates DNA-PK phosphorylation, which phosphorylates and inhibits HSP90. HSP90 is required to fold AMPK, so this causes AMPK downregulation, which leads to a metabolic disorder. So the summary is age-dependent DNA damage upregulates DNA-PK signaling, which phosphorylates HSP90 alpha. Phosphorylation of HSP90 alpha by DNA-PK reduces its ability to bind client proteins and fold them, resulting in their downregulation. One critical HSP90 client is AMPK and liver kinase B, and it's downregulated by DNA-PK-mediated HSP90 phosphorylation. This results in mitochondrial biogenesis defects and metabolic decline with age. So I hope you guys enjoyed this paper, and I hope you can appreciate the implications this could have on research into aging. And please comment any questions or anything you have. Thanks for watching.